informatic study described in the passage relies on the level of protein structure stabilized. So now I have to read Sag. Okay, let me zoom into my face so you guys can't see my messy room, bruh. It's me getting distracted. Let's see what we want. Let's put some. Color grading. Look at this. Color range. Acidification of the trypanosomal endosome induces APOL to transfer from aqueous lumen to integrate into the endosomal membrane, forming a closed ion channel. As endosomal membrane is recycled back into the membrane, the pH neutralization of the cyst side then activates the ion channel, allowing sodium and potassium cations to flow through. In other words, APOL1 acts as a ligand-gated ion channel that opens when its ligand H+ unbinds. Depolarization and dysregulation of ion flux then leads to osmotic lysis of the trypanosome parasite. To structurally characterize the topology of the APOL1 ion channel, researchers use bioinformatic methods based on APOL1 primary sequence to predict transmembrane regions as well as. So let's start with the fact that we're predicting transmembrane regions. That clearly uses hydrophobic interactions. Tell me I'm lying, bruh. As well as an experimental technique called the substituted cysteine accessibility method. I have never heard of SCAM, to determine extracellular and intracellular localization of the intervening loops. To perform SCAM, the research substituted specific amino acids in these loops as with cysteine residues and allowed the APOL mutants to form functional ion channels. A 53 kilodalton thioreactive reagent was then added to the cyst phase of the channels, and the molecular weight of the APOL complex was analyzed by non-reducing SDS pH followed by Western blot. This experiment was repeated using MPB without step tabulating. Okay. So, it's non-reducing. I know 2 is the answer. I mean 3. I think peptide bonds. I was wrong. It's just peptide bonds. Nothing about the hydrophobic, which makes sense because the question asked for the experiment and that information was presented before the experiment was presented. So next, based on the passage, the inclusion of which amino acid is most likely responsible for changing localization upon pH manipulation. options, guys. So we have isoleucine, which is nonpolar, glutamate, which is acidic polar, histidine is neutral, I believe, and lysine is, wait, is histidine neutral? Histidine, aka, so yeah, histidine is neutral, and then lysine is basic, right? We're trying to find the one that's most likely responsible for changing localization. So we have two neutral ones, meaning that it can't be the neutral ones. And then we have an acidic one and a basic one. Does this say anything about changing? It says pH neutralization, then activates the ion channel. So we're neutralizing. It's acidic. Where did I get acidic? Okay, yeah. Then we're gonna say. No, we're looking for the acidic one, so we're gonna say glutamate. That took a lot. I got it right, but that took a lot. Based on the data presented in the passage, which of the following best describes membrane protein topology of a POL1 channel? Based on the data presented, so we have 3 Western blocks. You have, they're all reactive with something. And then we have to find out where it's located, either on the extracellular side or the systolic side. I don't know, guys. If anything, we could say wild type and G are similar. So like G being different would be the answer. Correct. I got it right because I'm smart. I have to say this stuff. I sound conceited, but I have to say it or I'm gonna crash out. Pause. Okay. This is why I need to record myself studying, because I get so distracted. What were the purposes of the experiment with NPB without strep Devon, and of the experiment with the blank and detergent? Yeah, we could say positive and negative, right? But we don't know. Without strep, is negative. Yep. The cysteine MPBS reaction is a redox reaction involving nucleophilic attack by deprotonated cysteine side chain. If the reaction were attempted at pH 7, approximately what percent of cysteine side chains would be deprotonated? I want to say 0. It was 10. And I could have gotten the answer if I used Henderson about Hashibad. Okay. Trypanosomes were transfected with a new ion channel, channel X. Channel X is found to open trypanosomes that have been treated with APOL once pH neutralization occurs. Similarly, channel X opens in trypanosomes that contain the nicotinic acetylcholine receptor. When treated with acetylcholine, neither pH nor acetylcholine open channel X in trypanosomes without these two. So channel X is best described as. So what do these things do? Acetylcholine is a ligand that opens the nicotinic thing, so it has to be ligand gated. But I was wrong. It's gated. I guess it's an ion channel, huh? Because it responds to changes in membrane potential. I guess it says once pH neutralization occurs. That's it. That's the only thing. Oh, it literally says a new ion channel. Well, it could be like that. I don't know. That's interesting. How do you know there's depolarization? Is it because it's acetylcholine? Okay. Which statement most accurately describes the binding of PBGN to HMBS? Assume binding is significantly faster than catalysis. Crazy. A graph. I'm gonna look at the graph for the answer. PBG. Wait, stop. Okay, PBG has a higher affinity for the wild type enzyme than for any other enzyme form. That's a lie. It has a higher affinity for K13. PB has a lower affinity for R, blah blah blah, than any other. No. What? Okay, here's my problem. I need to know Henderson, Michaelis-Menten equation. From what I remember from Michaelis-Menten, I think Km is at the bottom. So the enzyme that has the highest affinity is crap. Okay, wait a second. Highest affinity is lowest Km, and the lowest Km is V. Lowest affinity is the highest Km, the highest Km is R. I got it right. The HMBS mutant variant is least likely to cause acute intermittent porphyria. Okay, let's ask ourselves, what is intermittent accumulation of the molecules? So I guess we want a slower or faster binding affinity. I feel like we would want something that reaches Vmax slower, so a bigger binding, a bigger Km, which is R. I was wrong, it's the opposite. Oh, it has to be something similar, closer to wild type. That makes sense. Because it's a genetic disorder. Okay, based on the data in figure 1, a mutation of which amino acid residue has the greatest impact on stability? From what I think, it's this one. And I was correct. Based on information in the passage, pyruvate in red blood cells can be further metabolized only by I gotta read. Phosphorylation. I saw pyruvate comment. This is how I I always just jump and then okay, A newborn girl is diagnosed with a rare genetic disorder that causes premature lysis of red blood cells through genome sequencing. Physicians identified a specific mutation, R510Q, in both copies of pyruvate kinase gene, which encodes the enzyme that catalyzes the final step of glycolysis. Pyruvate kinase produces pyruvate and ATP from PEP and ADP. Pyruvate kinase is a kinase, which means what? It phosphorylates. I'm locking in, it's phosphorylation. I was wrong. I love that for me. Pyruvate in red blood cells. That's the problem, guys. In red blood cells. Okay, because red blood cells lack mitochondria, pyruvate in erythrocytes does not enter the citric acid cycle in the electron transport chain. Therefore, red blood cells rely almost exclusively on glycolysis to produce sufficient ATP for cellular functions. In patients with pyruvate kinase deficiency, a lack of ATP is believed to compromise the integrity of the red blood cell membrane, leading to premature lysis in the spleen. Although these observations are well established, the exact mechanism by which low ATP alters the cell membrane is unclear. So the answer was reduction to form lactate and NAD+. And as you can see, because it only does glycolysis and doesn't do citric acid cycle or electron transport chain, it would be lactate dehydrogenase. Like, that's key, obviously. So that's why the answer is the lactate. I'm saying the milk, the lactate. Okay. Okay. Given a pyruvate synthesis is generally unidirectional. Unidirectional. Which of the following statements regarding the thermodynamics of reaction 1 is most accurate? So it's unidirectional and it releases ATP. If it creates ATP, that means what? It releases energy. Which means it has a positive entropy. Which means that it's spontaneous, which means that delta G is negative. So the net free energy change is negative. Correct. Based on figure 1, which of the following statements is true regarding pyruvate kinase as compared to wild type pyruvate kinase? Figure 1 is a graph with lines, if you guys want to know. Okay. Wild type and the pyruvate kinase have approximately the same Km in all conditions, but with varying Kcat. Oh, this is hard to look at, guys. I wouldn't say in all conditions. Oh, wait. It's not in all conditions. to remember what KCAT is. Oh, I was wrong. Which is why I need to know what KCAT is. Oh, I knew that. Okay. The information in the passage in the data in figure 1 support which of the following conclusions about ATP as an inhibitor? It is an inhibitor. That's the answer. Done. It definitely binds. It binds both free pyruvic kinase and enzyme substrate complex. Okay. Scientists predict that gene copy number is proportional to protein expression for pyruvic kinase. If this hypothesis is correct, which kinetic parameter would be expected to double when the gene copy number doubles? Okay, we're looking at protein expression. How does protein expression change kinetics? Let's think about that. I'm gonna say the equilibrium constant. It was maximum velocity, bruh. Okay. Oh, we're doubling gene copy and not expression. I think. Or maybe that's the same. What just happened? I thought I said 50 minutes. It's been 50 minutes. That's crazy. That sounds like a break to me. I shall be back. Okay, I'm back. Let me see something. Why is it keep going to 30 when I say 50? It looks like I'm not supposed to stay for 50 minutes straight. Or so. Okay. Let's go back. Based on figure 1, if a single epinephrine molecule binding to its receptor results in 10 active pKa molecules, 20 CAMP molecules, and 100 active glycogen phosphorylase B kinase molecules being produced, the signal amplification induced by pKa phosphorylating is typical target would be so it starts with 20 signaling molecules, 10 pKa active. It technically starts with 20 molecules and then it ends with 100 over 10. It's after? Okay. That's cool. Assuming that 2 kilograms of skeletal muscle are exposed to conditions that cause the cleavage of 100 millimole of glucose monomers per kilogram of muscle, and that 90% of these monomers are released from glycogen by glycogen phosphorylase, what is the total amount of unphosphorylated glucose released in the cytosol by this process? It's the 10%. Am I crazy? We'll start with how we have 2 kilograms of skeletal muscle, and then we have 100 millimoles per How are we supposed to know how many are unphosphorylated? Anyway, 100 millimoles per kilogram of muscle. So that is 200 millimoles, right? And then there's a situation where it's number one, 90%. And then we also don't know what's phosphorylated and what is unphosphorylated. So what is 90% of 200 millimoles? Like 180? So is it 180 or is it 20? We're gonna say 180. It was 20. I knew it was one of those. Okay. Each of the individual residues released during glycogenolysis can eventually enter glycolysis. Residues that are released by glycogen phosphorylase enter glycolysis after phosphoglucomutase converts from glucose 1-phosphate to glucose 6-phosphate. The remaining residues enter glycolysis when hexokinase converts them from glucose to glucose 6-phosphate, which figure shows the correct comparison of ATP consumption and production when 5 residues enter glycolysis, starting as Okay, so starting at glucose, we know that it consumes 2 ATP and produces 4. So that only got rid of one option. I fear. Okay, the remaining interglycolysis and hexokinase. What if I, hear me out, cheat, and look at glycogenolysis? I want to say glycogenolysis uses less ATP. But it produces the same amount of ATP because storage. Oh, I was right. Like, logically speaking, that makes sense. Okay. The skeletal muscles collectively contain more than twice as much glucose stored in glycogen as found in the liver. Compared with the liver, the amount of glucose released into the blood from the muscles is likely. Okay, skeletal muscles have more than twice as much glucose in the form of glycogen than liver. Compared with the liver, the amount of glucose released in the blood from the muscles, it has to be less. Because they're focusing so much on storing that. I want to say, yeah, it was less. It was less in both. Did I put that as an answer? No. But did I know that was the answer? Yes. I always second guess myself and choose the wrong answer. So yeah, it releases less because it needs that energy. Your muscles be muscling. Based on Table 1, the D187V form of GALC resulted from what type of nucleotide change? Okay. Let's see. Can I see first? Do I have to? Oh my gosh, I have to read it. I'm sorry. Homeostasis of GC is maintained by its dynamic synthesis and degradation, as shown in Figure 2. GC is synthesized by the enzyme ceramide galactosyltransferase using UDP. Okay, guys, UDP is uracil. I was like udenosine. Uracil diphosphate. Galactose, UDP, and ceramide are substrates. It is degraded by the enzyme galactocerebrosidase, which hydrolyzes the glycosidic bond to release galactose and ceramide. A deficiency in GALC activity leads to a buildup of GC. And prior research has connected this buildup to neurodegeneration. Okay. See, it literally says based off of Table 1, and I was like, oh, I have to read. I could have just went to Table 1. Based on Table 1, D187 resulted from what type of nucleotide chain? It says A to T, right? A is a purine and T is a pyrimidine. So it's purine to pyrimidine. Let's lock in. 90% of people got that right. And I'm one of them. What is the stereochemical chemical configuration of galactose component of GC. Now, hear me out, guys. Before I even try to think, we're gonna say that it's found in the body. And what's found in the body? Else. I was wrong. Oh, I was thinking L amino acids. So it's L amino acids and D sugars. Okay, we got that for later. Sugars are in D and amino acids are in L. Okay? Yeah. After galactose is released from GC and linearized, galactose contains all the following functional groups except. Okay, so it's linearized. In this situation, you would have to know how galactose looks. Thankfully, I don't. It's like that. Let's see. There is definitely secondary alcohol. I can see a ketone being present. A primary alcohol would be crazy. Like if there's a ketone, there's no primary alcohol. There's no ketone. That was the answer. The answer was there was no ketone. Oof. That was a big oof. Okay, let's see. Based on the information in the passage, which bond is cleaved by gal C? Based off the information in the passage, which one is cleaved by gal C? It's the bond between galactose and ceramide. But is it carbon 1 or carbon 4, guys? We're gonna say carbon 1. Oh, we're right. Okay. Each of the following amino acid residues is likely to be found in the portion of RRM that binds blank except. Okay, so before we even read the text, why am I? Okay, before we even read the passage, we're gonna identify these amino acids to see if there's one that just really sticks out. So we have leucine, which is nonpolar. Arginine, which is basic polar. That's a plus. Glycine, nonpolar. Serine. I'm pretty sure serine is neutral. So, to be honest, none of them stick out, but it's probably not leucine or glycine. But one is charged, which is arginine. But we're gonna definitely double check. So translation of mRNA to produce proteins is highly regulated. For example, one factor that regulates eukaryotic translation is the polyadenylate binding protein, PABP upregulates translation by binding the poly A tail of an mRNA molecule to help recruit the ribosome. PABP consists of single polypeptide chains and contains 4 distinct RNA recognition motives, each of which contains 4 beta strands and 2 alpha helices. Mutation experiments revealed that each RRN binds poly A tails through residues in several beta strands. Crystallography showed that the two alpha helices in each RRN, which consist of amino acids 9 through 24 and 52 through 60 respectively, help hold the beta strands in correct orientation for RNA binding. In addition to binding poly A tails, PABP can also bind to eukaryotic initiation factor E through the alpha helices in the RRN region. When both blank and mRNA with a poly A tail are present with PABP, a ternary complex between three molecules can form. This complex further enhances translation relative to the complex alone. To better characterize these binding interactions, PABP was electrophoresed in native gel in the presence of mRNA containing a poly A tail. When that was added, PABP affinity for poly A tails increased tenfold. Additionally, observations revealed the affinity increased 12-fold when mRNA with the poly A tail was added. So, look, it didn't even say really anything about other amino acids, which is why you don't have to read for all of them. Each of the following amino acids residue is likely to be found in a portion of RRN that binds blah blah blah, except. So, the portion of RRN that binds I'm gonna just say, oh, it was glycine, guys. Oh my gosh. How did I not know that? What? Okay, given the PABP is primarily found in the reducing environment of the cytosol, the information in the passage best supports the conclusion that the RNA binding beta strands are held in the correct orientation for binding by let's say covalent interactions by the backbone. It's non-covalent. How are non-covalent interactions that powerful? The tertiary structure of a protein is three-dimensional form. Tertiary structure is primarily stabilized by non-covalence between the side chains. And then this also said that it's a tertiary structure, so that's how you find the answer. Based on the information in the passage, which of the following best describes the order of formation to the ternary complex? Okay. This is when drawing out the system while reading is important. So, let's see, what starts first? Okay. Which of the following best explains why researchers use native gels for electrophoresis experiments to preserve the three-dimensional structure? Yep. That's why. I didn't even look at the other options. I'm like, yep, that's the reason. That's gonna be my downfall someday. Which of the following descriptions of the interactions between PABP, blah blah blah, and poly A tail of mRNA is best supported by the passage? Both the PABP and blah 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 bind poly A tails directly to increase translation levels. Formation of ternary complex exhibits cooperativity. Oh, I knew that. Anyway, That's what I got. 43%. Next. Scientists studying evolution in yeast found that a certain species underwent a reduction in chromosome number with no loss of coding information. Given this, which of the following mechanisms best explains how this reduction in chromosome number may have occurred? Okay, from what I know, every time DNA, like a cell, divides, the telomeres get shortened. So my thought is the telomeres get shortened. Okay, wait a second. End-to-end fusion of 2 chromosomes followed by an activation of 1 centromere on the newly formed chromosome, breakage of a chromosome at the centromere, and fusion of 2 resulting chromosomal portions to the ends of other chromosomes, transfer of DNA sequence from one chromosomal arm to a different chromosomal arm. I think is 1 and 2. And I was correct. So basically in this case, end-to-end fusion of 2 chromosomes followed by an inactivation of 1 centromere on the newly formed chromosome is how you keep DNA during reduction and breakage of a chromosome at the centromere, and fusion of the 2 resulting chromosomal portions to the ends also preserves DNA. Based on the pKa of histidine side chain given in the passage, which of the following best describes the charges of the his dipeptide side chains at pH 1? So the side chain of histidine is approximately 6, which is giving neutral, right? So the side chain is 0. But it does have a small percentage of a positive charge. That was great. What is the molecular geometry of nitrogen atom in a peptide bond that leads to 2 histidine residues? In the side chain? Based off of this, it's tetrahedral. It's trigonoplanar. Oh, there's a double bond. Okay, I tried. Okay, wait a second. The redissolved mixtures were most likely subjected to which of the following techniques to separate his from monomeric histidine and generate the data in table 1? Gel electrophoresis with Kamazi staining, high performance liquid chromatography, spectrophotometry at 280 nanometer wavelength, and northern blot and denser analysis. Okay, northern blot is RNA, so you can get rid of that. Gel electrophoresis and Kamazi staining. I would say yeah. If not, it's spectrophotometry. Okay, it was high performance liquid chromatography, but most of the people that answered this thought it was also gel electrophoresis. High performance liquid chromatography is a technique in which the mixture of molecules enters a column packed with beads. Okay. Yep. That's all I need to know. Which of the following observations would provide additional support for the hypothesis that his formation was critical to prebiotic protein formation? A dipeptide consisting of non-histidine amino acids increases the rate of polyglycine formation. No, that's opposite. Monomeric histidine does not increase the rate. That is support. So number 2 is support, which basically just leaves us with C because I got rid of 1. His increases the rate of formation of peptides other than polyglycine. It's the other option. As the initial pH of the reaction that forms his increases from 5 to 7, the yield of his increases by a factor of approximately 0.31. It's 3.3. Oh, you do it the opposite than what I just did. A certain eukaryotic organism has multiple distinct genes with highly similar sequences that are expressed at different times in the organism's life cycle. These genes could have risen by... Okay. They have multiple distinct genes with highly similar sequences that are expressed at different times in an organism's life cycle. I personally think that this might be alternative splicing, but it was gene duplication. 59% of people also thought it was alternative splicing, so I think that I'm not stupid. Genes with similar sequences are considered evolutionarily related. In other words, they are believed to have a common origin in a single gene from an ancestor organism. During the process of molecular evolution, portions of a chromosome can be duplicated by mechanisms such as unequal crossing over. Over time, duplicate genes can undergo mutations, causing them to have similar but not identical sequences. These differences allow the genes to carry out distinct roles at various times in an organism's life cycle. The question states that some organisms have multiple genes with highly similar sequences. These similar genes most likely arose by gene duplication. I had never even heard of gene duplication, to be honest. Okay, I need to add these words to my list though. Because there's stuff that I don't know. Let's put it under I should make a central dogma cheat sheet and put it under but we got alternative splicing. So that alternative splicing has the same gene, by the way. That's why we were wrong. Conjugation is the other one, and that's between prokaryotes. It does not occur in eukaryotes. And then the other option was transformation. And transformation also occurs in prokaryotes, not eukaryotes. Based on figure 1, substitution of cytosine for which of the following nucleotides would cause the loss of function mutation? Okay. Cytosine. indicates mutation resulting in truncated protein. Substitution of cytosine for its complementary base. So based off of the caption for the figure, it says that it was substituted for its complementary base. So the complementary base of cytosine is guanine. So we would have to put the structure of guanine, and I would have to remember the structure of guanine. And if it's an RNA or DNA, because they have a ribose sugar and they have a deoxyribose sugar. So I'm going with is a deoxyribose. I was wrong about the structure of guanine, but I was correct about it being deoxyribose. So guanine is the one with the ketone. Okay, let's try this again. We're studying for 50 minutes. Confirm. It still didn't do it. It says I'm studying for 30 minutes. It said, girl, take a break. And I will. Just not right now. A scientist studying the sequence of the LPL homodimer would most likely expect that ribosomes must translate a total of 1,350 mRNA nucleotides into amino acids to form one 100 kilodalton LPL homodimer. Okay, let's see how we get this stuff going. Okay, we're about to read the passage. Because I don't see any numbers in the figures. So, abnormal blood lipid levels play a significant role in the development of coronary artery disease. The discovery of genomic variations that confer protective effects against CAD has prompted investigations into biological impact of these protective alleles. Researchers have observed that specific single-base variations in the ANGPTL4 gene are common among patients with a low occurrence of CAD. ANGPTL4 is located on the chromosome 19 and codes for angiopoietin-like 4, a multifunctional glycosylated protein. One of the functions of angiopoietin-like 4 is the regulation of lipid metabolism. Specifically, angiopoietin-like 4 inhibits the activity of the enzyme lipoprotein lipase. So LPL is lipoprotein lipase. Generally, active LPL is a homodimer consisting of two individual subunits that are each approximately 450 residues in length and weigh 50 kilodaltons. So if we were about to go off of that and it's a homodimer, we would say that it is 100 kilodalton homodimer. And as for mRNA, I don't know. Because 450 residues plus 450 is like 900, I think. 900. But what is a residue? Is it 900 times 3? Why, you ask? Because 3 nucleotides code for an amino acid. and one of these answers is 2,700 mRNA nucleotides. So we're gonna go with that, and I was correct. Yup, my reasoning is also correct. Okay. Based on Figure 2, ANGPTL4 mutations were found to decrease CAD risk by significantly altering which of the following? Okay, so we're going to look at Figure 2. And we want a decrease in risk, right? Which is LDL. I'm pretty sure LDL is bad and HDL is good, right? So a p-value of 0.05 is good. So I feel like we can only say which is HDL cholesterol and but it says decrease. Altering which of the following? I think it's three only. It's only the triglycerides. Oh, I looked at the number wrong. Okay, so I was right. 0.05 is a good statisticalness, is a good p-value, but I thought one of the values was under it and it was over. So that's why I got it wrong. The knockout mouse model and constitutively active mouse model of ANGPTL4 are best represented by, okay, so we didn't get that far. To identify common loss of function mutations in ANGPTL4 DNA samples from 43,000 individuals with CAD and 78,000 individuals without CAD or genotypes. A detailed analysis of ANGPTL4 genomic variants was performed, and the results of the individuals without CAD were mapped in Figure 1. The lipid profiles of ANGPTL4 mutations non-carriers were also compared to those of mutation carriers, and that's what Figure 2 was about. So to further analyze I literally could have just looked at Figure 3. I'm in Table 1. I didn't have to read. To further analyze the effect of ANGPTL4 on blood lipid levels, ANGPTL4 knockout mice and mice that constitutively express ANGPTL4 were created. Researchers hypothesized that 2 of the 4 biological profiles shown in Table 1 would be observed in these mice. So let's think about this again. The knockout mouse model and constitutively active mouse model of ANGPTL4 are best represented by. Okay, so let's think. So this model, the knockout mice, means that they do not have ANGPTL4. And there's 2. So profile 1 and profile 3. So it's already like I can't even get these options away by doing that. So you just have to know if it increases or decreases lipoprotein lipase and serum triglyceride levels. Do these mice have high cholesterol? I feel like that's the only way I can figure. let's say that we want more lipase activity and less triglycerols. So we're going with profile 3. And then 3 and 4. And I was correct. Okay. The graph below shows messenger RNA expression levels for JARID2, a protein known to regulate gene expression during embryonic development, and 2 of its target genes, OCT4 and SOX2, in cattle, Bos Taurus embryos. Why do I feel like I'm speaking so much? At the 2-cell stage, these embryos have been either treated with small interfering RNA, which is siRNA, targeting JARID2 mRNA, or left untreated. Okay. Which of the following? I did not have to read all that. And maybe that's my downfall. Which of the following most likely explains why expression of OCT4 and SOX2 mRNA was higher in the siRNA treated embryos? Okay. So here are the options. OCT4 and SOX2 are essential for JARID2 gene expression. I would say that is not correct. Because if it was essential, well, actually, let's leave that. Translation of those two occur more rapidly when the gene expression is lower. We can't say that because we're not given that. I'm gonna say that the JARID2 normally increases OCT4 and SOX2 transcription at the 2-cell state. I was wrong. It decreases the transcription. I do need to figure out this psycho situation. I know that's something I lack in. For sure. Like gastrulation, blastulation, nodotube situation, notochord is what it's called. Anyway, based on the passage, so basically that means I have to read hepatocytes, which are liver cells that are producing BHP, are also most likely to have. Okay. So although the ketone body beta hydroxybutyrate, which is BHP, is commonly associated with the pathological condition diabetic ketoacidosis, its most recognized physiologic function is an alternative fuel source produced by the liver when glucose is sparse. More recently, evidence has emerged that BHB also serves as a signaling and regulatory molecule that is associated with protection against the effects of aging, inflammation, and neuronal excitotoxicity. BHB released from the liver is taken up by muscle, brain, and other peripheral tissues where it is trafficked to the mitochondria. Okay. BHB is reversibly converted to acetylacetyl-CoA, which can then be processed through beta oxidation. Because BHB is processed in the mitochondria, the systolic NAD plus pool is preserved. In addition to its use of redox cofactors, systolic NAD plus also acts as the substrate for the sirtuin and poly-ADP ribose polymerase PARP enzymes. By preserving the systolic NAD plus pool, increased BHB levels enhance the anti-aging and anti-apoptotic effects of the sirtuin and PARP enzymes. BHB acts directly as an agonist for several receptors, including hydroxycarboxylic acid receptor 2, which is HCAR2, a G-coupled G-protein-coupled receptor, GPCR, that upon activation inhibits adenocyclase and its downstream effectors. In adipocytes, BHB-mediated activation of HCAR2 reduces lipolysis, decreasing serum levels of pro-inflammatory free fatty acids. In the colonic epithelium, HCAR2 activation is critical in the maintenance of gut membrane integrity. In this way, BHB acts similarly to the short-chain fatty acids, for example, butyric acid, produced from fermentation of dietary fiber and protecting gut health. Several mechanisms have been proposed to explain BHB's action as a neuroprotectant against excitotoxicity. In glutamatergic neurons, BHB inhibits vesicular glutamate transporter 2, which is VGLUT2, inhibiting the packaging of the excitatory neurotransmitter glutamate. In GABAergic neurons, BHB infusion stimulates production of the inhibitory neurotransmitter gamma-aminobutyric acid, which is GABA, and decreases alpha ketoglutarate levels. Okay, so from what I'm seeing here, BHB becomes acetoacetate, which becomes acetoacetyl-CoA, right? So it seems like it would upregulate the creation of those two enzymes, but that is not an option, by the way. So it's not upregulating glycogen synthesis, but instead upregulating fatty acid synthesis. From what I remember. It does downregulate phosphorylase kinase activity, but it also downregulates phosphofructose kinase 2, which is in Okay, that's a lot. So to answer this, I would have had to possibly draw the entire situation. What is upregulated and what is downregulated. That's a lot. Okay. The second reaction shown in figure 1 involves an enzyme that transfers coenzyme A from succinyl-CoA to acetoacetate. If this enzyme were used in the citric acid cycle in place of succinyl-CoA synthase. CoA synthetase. The overall yield of high energy molecules in one round of citric acid cycle would be decreased by 1 NADH trust because the citric acid is KISS. I'm sorry. It was actually decreased by 1 NTP. I knew that. Anyway, bro, I'm ready to clock out. Glucose and BHB can each be converted to 2 acetyl-CoA molecules. How many NAD plus molecules, both systolic and mitochondrial, are consumed by conversion of glucose to 2 acetyl-CoA and conversion of BHB to 2 acetyl-CoA? So we know 2 NAD plus, yes, are consumed per glucose. Now the answer is how many are consumed for fatty acids, and I think is 1. And I was wrong. It's actually 4, but most people thought it was what I said. Oh, that makes sense. I forget that glucose separates. Okay. The effect of BHB on adipocyte lipolysis is an example of, okay. Negative feedback because it inhibits. We don't know how. And GABAergic, that's not what it says. GABAergic. It says GABAergic. Neurons. Glutamate can be converted either to GABA or alpha-ketoglutarate. Which statement would best explain the effect of BHB on this equilibrium? Okay. Acetoacetate, produced from BHB oxidation, is the alpha keto derivative of GABA and competitively inhibits transamination of alpha glutamate. I don't think that's correct. BHB is gluconeogenic substrate that is metabolized into pyruvate. No, I'm not even going to finish that sentence. BHB acts as an allosteric inhibitor of glutamate decarboxylase, the enzyme that catalyzes the decarboxylation reaction. That's not correct. That doesn't sound right. Acetyl-CoA produced from BHB metabolism temporarily depletes oxaloacetate, which acts as a co-substrate for the transamination reaction. I'm gonna go with that one. The other ones just didn't sit right. And guess who got it right? Me. Other ones sound crazy. Okay, next question. Stenosis is the abnormal narrowing of a tubular structure in the body, such as a blood vessel. Stenosis of the afferent arterioles is the kidney. Would be expected too. Okay, let's think about this. Are afferent going away or tube? Because it's afferent, I'm just gonna say it's going away. If we are narrowing, if we are narrowing that tube, these are blood vessels, and they're going to the body. pressure decreases, then the glomerular filtration rate decreases. I got it right. That took a lot of brain power. Okay. We got it right though. Oh my gosh. The effects of glucocorticoid deficiency are known to include impairments in superficial wound healing, memory formation, and other physiological phenomena. That's crazy. Guys, manage your stress. This deficiency most likely leads to these wide-ranging effects because, so no glucocorticoid. Glucocorticoid, from what I know, creates aldosterone and cortisol. So it's less aldosterone and less cortisol. Less cortisol, from what I know, is good. But less aldosterone. What does aldosterone do? Aldosterone is responsible for the NAK plus pumps in the nephron, and it pumps 3 and 8 plus into the nephron. Its receptors for glucocorticoid hormones are expressed in nearly all types of cells in the body, and that's why. So they're found in the adrenal gland, they're found in the liver, and they're found in skeletal muscles. Oh my gosh, that's a penis. I'm sorry. You just have a picture of a penis here. The neuroendocrine signaling cascade that induces adrenal hyperplasia in patients with 21-hydroxylase deficiency involves coordinated activity among which organs? Okay, let's go ahead and read this before wait a second. The hypothalamus communicates to the parathyroid gland, which communicates to adrenal glands. So, based off of this, I don't even have to read. Actually, I do. Bruh. Patients with CAH exhibit hypoglycemia due to numerous endocrine abnormalities. In healthy individuals, hypoglycemia triggers an increase in serum level. Okay, hypo is low glucose. If you have low glucose, your glucagon is going to go up. So we know that option is correct. Now, if your glucose is going up, your epinephrine is going to go up. Glucose and insulin are opposites, so you know that's not the answer. Boom. Okay, glucagon is the alpha cells because glucagon has an A in it, and insulin doesn't. That's gonna be my reasoning for knowing glucagon is produced by alpha cells. What changes in blood volume and serum sodium content would be expected as a result of using synthetic hormones to replenish aldosterone in patients with CAH? Okay, I didn't read this, but we're gonna go off of like brain power. Aldosterone is responsible for the Na plus K plus ATPase pump, right? And if we're doing that, we're increasing blood volume and increasing serum sodium content, because what? Water goes where the solute is. Correct. It's 3 Na out, and I said Na in. But other than that, it's correct. A 10-day-old male who was not feeding well was diagnosed with 21-hydroxylase deficiency despite having normal physical examination results apart from signs of dehydration. Wow, this is sad. Which of the following best explains why physical examination results can be largely normal in males with this condition? Okay. The testes compensate for abnormal adrenal function by synthesizing glucocorticoids. That sounds like it could be correct. I don't know the answer. The Y chromosome contains extra copies of genes mutated in CAH. That's like the opposite, I fear. High testosterone levels lead to abnormal sexual development earlier in females than in males. I don't know about that one, because this is a 10-day-old. How much testosterone does a 10-day-old have, you know? Only female sexual differentiation is dependent on serum glucose. I'm going to go with the testes. It actually was that one. It sounded crazy to me. But I guess 10-day-olds have a lot of testosterone. Like, I knew it causes changes in women more than men, but it's just, I don't know. Given that the function of male and female gametes results in fertilization, which statement best explains how unequal crossing over during gametogenesis led to 21-hydroxylase deficiency in the fertilized embryo? One of the parental gametes. Let's see. So we have this gene on chromosome 6. That's not there. So we're gonna say that it was deleted, and I was correct. A student analyzing dividing cells of a certain type under a microscope finds that each daughter cell produced contains half the number of chromosomes as its parents. Given this, cells of this type would most likely contain all of the following except, so we have peroxisomes, nuclear poroprotein, single-stranded genomes, microtubules. We know single-stranded genomes is it. Microtubules are found when they separate, and that was used. Peroxisomes sounds very advanced. It was single-stranded genomes. Because single-stranded, basically it's saying since the daughter cells were haploid, it was eukaryotic cell, and single-stranded genomes are found in prokaryotes. One of the sperms reaches the zona pellucida during fertilization. The cortical reaction is triggered. This aids fertilization by... Okay, wait a second. Okay. It reaches the zona pellucida during fertilization. The cortical reaction is triggered and aids in fertilization by preventing more than one sperm. Yep. I remember that one. Okay. We have more incomplete questions. Okay. Which observation would not support the hypothesis of gene transfer from eukaryotes to prokaryotes described in the package? So we have to. Looks like we're starting in 30 minutes. We're still on this one test. Okay. Eukaryotic organelles such as mitochondria and chloroplasts are believed to be relics of formerly free-living prokaryotes. The transition from hypoxic to an oxic atmosphere is said to have enabled primitive eukaryotic anaerobes to engulf ancient aerobic prokaryotes and consequently acquire the ability to produce energy through oxidative phosphorylation. This endosymbiotic theory of eukaryotic evolution also postulates that endosymbiosis resulted in larger eukaryotic genomes, which originated from the partial transfer of mitochondrial genes to the nuclear genome. I zoned out for a second, I'm not gonna lie. Wait a second. An integration into the host genome, mitochondria-derived genes became undistinguishable from the original nuclear genes. Okay. Researchers have alternatively proposed that after a prolonged period of symbiosis, the possibility exists of gene transfer from eukaryotes to prokaryotes. This hypothesis was initially supported when copper zinc superoxide dismutase, a metalloprotein confined to the cytosol of eukaryotic cells, was found in Photobacterium leognathi. The free-living bacterium P. leognathi is also a known symbiont of ponyfish, a small fish species native to the Indian and Pacific Oceans. SODs are antioxidant enzymes that serve as the cell's first line of defense against reactive oxygen species. ROS produced by the electron transport chain damage proteins by oxidizing amino acid residues and metal ions on prosthetic groups, but can accumulate during times of biochemical and environmental stress. Superoxide radicals, a form of ROS, are sequestered by SODs and converted into less toxic hydrogen peroxide and O2 gas, which observation would not support the hypothesis of gene transfer from eukaryotes to prokaryotes described in the passage. So that basically they're saying the gene transfer occurred because there's similarities. So we can get rid of discovering no difference between the gene sequences of P. leognathi and ponyfish. Discovering P. leognathi and ponyfish SODs have similar amino acid sequences in their non-catalytic domains. That doesn't help. Discovering copper, zinc, SODs, and other free-living bacterial species with no known eukaryotic symbiotic host. That's the only option that says, oh, there's a difference. And that's correct. Maybe. SOD mRNA in P. leognathi encodes a signal sequence that directs the transport of SOD protein for secretion. This signal sequence will direct SOD proteins to which structure in the P. leognathi. Okay, so it encodes a signal sequence that directs the transport of protein for secretion. What does that? The Golgi body. The Golgi body is the sorter. Wrong, it's a plasma membrane. I was thinking it was a plasma membrane too. Given this information, which of the following would most likely result as a consequence of toxin exposure? The daughter cells with multiple copies of the gene, daughter cells containing the same copy number of the gene, delayed separarization during binary fission, non-disjunction, and somatic. I think it's non-disjunction. Yup. Because the microtubules can't pull any apart. Yup. Okay. Investigators use a ribosome profiling technique to measure the level of SOD protein synthesis in P. leeuwenhoekii in ponyfish cells during ROS accumulation. After introduction of ROS, investigators found that P. leeuwenhoekii cells accumulated ribosome or SOD mRNA complexes at a faster rate than did ponyfish cells. What is the best explanation of this result? So let's go back to ROS. ROS oxidizes amino acids. Great. So if the leeuwenhoekii cells accumulated these complexes faster, it means that they had less of the ROS, like less effects. So the leeuwenhoekii SOD mRNA is capable of being bound by more than one ribosome, maybe. P. leeuwenhoekii SOD mRNA has a greater affinity for ribosomes than ponyfish mRNA. Maybe, because if it has a greater affinity for the ribosomes, it gets translated before it can die, you know? mRNA transcribed by the ponyfish SOD gene undergoes immediate degradation. mRNA transcribed by the bacterial SOD gene is being translated simultaneous. Okay. That's the answer. Simultaneously, because it's bacterial. That was so easy. Because a ponyfish is eukaryote. Okay, anyway. In anaerobic environments, P. leeuwenhoekii can produce energy by using an inorganic final electron acceptor other than oxygen in the electron transport chain. Under anaerobic conditions, which of the following is active in P. leeuwenhoekii but is not active in ponyfish cells? So we know glycolysis is active. Fermentation is also active in eukaryote cells. Gluconeogenesis? Synthase. I had a feeling. Okay. The glucagon-induced entry of glucose derived from liver glycogen into the bloodstream for transport to other tissues involves close interaction among which enzymes? Okay. Glycogen phosphorylase debranching enzyme and glycogen synthase. Glycogen phosphorylase debranching enzyme phosphoglucomutase glucose 6-phosphatase. Compared to the response that occurs during absorption of dietary carbohydrates, the response of glycogen particles to glucagon released during post-absorptive conditions is likely a result of. So, glycogen particles to glucagon. Glucagon is like, oh no, glucose is gone, so we're gonna have to break down these glycogen stores. So, an increase of incorporation of phosphorylated glucose molecules into glycogen molecules is no. An increased rate of phosphorylated glucose released from glycogen particles sounds about right. Decreased rate of glucose released from non-reducing ends of glycogen branded. No, we need more glucose because it's glucagon. An increased rate of phosphorylated glucose released from reducing ends. 6-phosphate. So released from glycogen particles or released from the reducing ends of glycogen branches. What's the answer? I think it's just glycogen particles. It's not branches because it's from the non-reducing ends. In addition to phosphorylation events described in the passage, glycogen synthase can be reversibly regulated by glucose 6-phosphate binding to carboxy terminal arginine residue outside the active site. Given this fact, which type of interaction presents allosteric regulation? Allosteric is like permanent. So, sorry. So it's covalently bound. Oh, but it is reversible. But this is allosteric. It's reversible. This is non-covalent. It's non-covalent. So I got it mixed up. I think non-covalent is allosteric and covalent is non-allosteric. Okay. That makes sense because it's reversible. During glycogen breakdown, if the average rate that each residue stored in liver glycogen particles is released as phosphorylated glucose is directly proportional to the particle surface area of volume ratio, which of the following statements describes the expected relative rates of glucose 1-phosphate released from individual liver glycogen? Isn't it the same if it's proportional? Oh, it's 1/10 as fast. I got the equation wrong. The researchers hypothesized that glycogenin is required for normal glycogenesis in all body tissues. Do the results presented in the figure 1 support this hypothesis? Let's look at figure 1. Figure 1 has liver and heart glycogen concentrations. GN question is it required for normal glycogenesis in all body tissues? The answer is no, because knocked out still has glycogen. The answer is no, because they were normal. This is bad, guys. This is bad. These are a lot of questions.